Hello, my name is Nettie LaBelle-Hamer, and I'm the Interim Vice Chancellor for Research here at University of Alaska Fairbanks. I'd like to welcome you to One Health, One Future 2021. I'd like to say a few words about UAF research before you get back to your important work. Here at UAF, about 80% of the research that we do is directly relevant to Alaska's people and Alaska's industries. So if you think about the initial work that we do is the center of the drop, then we first have a technology impact, and we can have a community impact, we can impact the entire state, or we can have a global impact, depending on the work we're doing and the collaborations that we have along the way. UAF research is directly relevant to Alaska's people and Alaska's industries, and it's evident across the whole university. Here's a list of the UAF's major research institutes, centers, and programs, and you'll notice that the Center for One Health is right there in the middle. It's a very important part of the whole that makes up the University of Alaska Fairbanks research enterprise. I'd like to talk a little bit about my view as a physicist of the One Health concept. I understand it came from veterinary med and the National Institutes of Health and bringing those concepts together. But to me, once it was brought together and presented in the way that you are working on it now, with human health and animal health and environmental health all working together and intertwined, it makes perfect sense. And I really appreciate what happens when you bring these elements all together. UAF's One Health mission is to work to improve our research capacity, more effectively promoting well-being in the North, to build teams to create research, education, and outreach programs around One Health, and to create long-term funding opportunities through collaboration. This is important, especially for us, though, here in Alaska. One Health concepts and the ideas lie at the core of Indigenous worldviews. Food and cultural security, energy, water availability, infrastructure, sustainable economy, health, and resilience are all part of the One Health concepts. The people's health, the animal's health, and the environment's health, all interrelated. And another important part of One Health is that it employs an interdisciplinary approach to problem solving, awakening what Scientific American has dubbed sleeping beauties of science. Now, I know this graphic is difficult to read on this slide, but I encourage you to go look at the Scientific American Graphic Science article in January 2016. That's where I got this picture. The vertical axis is time, starting in 1900 up to 2010. The colors indicate the discipline, the science discipline. There's 4 represented here, different colors. Physics is green, chemistry is purple, for example. And the black dotted at the beginning, or at the lower time, is when a concept first was introduced into a discipline. And then as it goes up in each of the years, it shows how many times that work is cited. And these are called sleeping beauties because if you notice, it's pretty sparse citations going up through the years until all of a sudden there's a blossoming that happens. And the blossoming that happens has been linked to when interdisciplinary approaches come into that idea. So when you have an idea that in one discipline and a scientist or a researcher or an engineer or a community member from another discipline comes in and looks at it differently, new ideas really bloom and blossom, and that's what you see in these sleeping beauties. One Health employs this interdisciplinary approach from the beginning, and it allows for this awakening to happen, hopefully faster than the 100 years that you see here. In addition, though, we know now in 2021, more than ever, the critical area of research, education, and outreach for One Health. With more than 70% of the emerging human diseases coming from the animal world, this is where we know for a fact through this pandemic that it's important for us to study all 3 aspects of One Health together. So I applaud you, I thank you, and I welcome you to Fairbanks. Thank you for participating. Even if the participation is virtual, we appreciate you being here. Thank you, and have a great meeting.
Thank you, Vice Chancellor Lavelle Hamer, for that introduction. I'd now like to introduce our keynote speaker for today. Dr. Emily Jenkins, PhD, DVM, BSc Honors, is a professor and acting head at the Department of Veterinary Microbiology at the Western College of Veterinary Medicine at the University of Saskatchewan, Saskatoon. She's previously served as a wildlife and disease specialist for the Government of Canada. She teaches in parasitology, public health, One Health, and emergency management. Her research takes a One Health approach to diseases that transmit among animals and people via food, water vectors, and the environment. Locally, she serves as chair of the Wildlife Health Research Fund and University Northern Studies Committee. More broadly, she is a Canadian representative to the Terrestrial Working Group of the International Arctic Science Committee, a member of the Alaska CDC and Native Tribal Health Consortium, One Health Working Group and Climate Change and Infectious Disease Working Group. She's involved in the international circumpolar surveillance of emerging infectious disease and assists with the Arctic Council. She also serves as associate editor of the International Journal of Parasitology, Parasites and Wildlife, and on the editorial advisory board of Food and Waterborne Parasitology. Please join me in welcoming Dr. Emily Jenkins. I'd also like to welcome and introduce the speakers that will come after Dr. Jenkins. Dr. Ann Zink, who is our Chief Medical Officer for the state of Alaska. Peter Rabinowitz from the One Health Research Center at the University of Washington, and Dr. Tom Hennessy, who is now retired from this CDC Center for Arctic Investigation in Anchorage, Alaska. Thank you all for taking the time to join us, and I hope you'll enjoy the discussions today. Remember that after the speakers, there'll be a plenary panel, and then some time to network, and then posters. So please enjoy this day, and talk to you soon.
Hello, I'm Emily Jenkins, and I'm speaking to you today from Saskatoon, which is in Treaty 6 territory and homeland of the Métis. And I wish we could all be together, but we can't, so this is the next best thing. And I'm here today to tell you a little bit about the Canadian Arctic One Health Network, which is a joint effort between myself, based at the University of Saskatchewan, and Patrick Layton at the Université de Montréal. And we have network investigators across the country in Canada, including Shirley Harper at the University of Alberta, who is taking the lead on some of our water security and safety issues, particularly in Nunatsiavut in the Eastern Arctic. Dr. Susan Coots, who you will have heard at this conference as well, giving a keynote from the University of Calgary, who is involved in the vectors and vector-borne disease aspects, people, dogs, and wildlife, as well as some aspects of food safety and security that involve wildlife. Nicolas Lecomte at University of Moncton, who is also based in Nunavut and doing research there on wildlife and vectors. Then also at the Université de Montréal, we have Dr. André Revel, who works on people, dogs, and wildlife, along with Cécile Anichelin. And that is where the focus of our efforts have been going on, also modeling Arctic fox rabies. And so as exciting as those two pieces are, I'm going to focus today on the vectors and vector-borne diseases and the food safety and security aspects of the network with which I'm more familiar. And our goals overall for this network were to establish sentinel sites across the Canadian Arctic. And these sentinel sites would hopefully involve standardized protocols done at roughly the same times of year, and not just include scientific measurements, but also local and Indigenous observations. And that is something that's expanding all the time. It began at sites where we were already working, where we already had established field work, but that is growing as we engage with more and more partners. The modeling component is primarily coming out of the Université de Montréal, and this is where we are developing and parameterizing epidemiological models, but then combining those with climate data in order to get an idea of what lies ahead for the Canadian Arctic, because it is very much not a static environment, very dynamic. And then finally, one of the bigger aspects of One Health that we want to focus on is mitigation, actually doing something, an action part of One Health. And there is a strong need, we recognize a strong need to build northern local capacity through training Arctic One Health scholars, through optimizing diagnostic testing for low resource remote environments where it may not have all the toys and machines and access, and enabling citizen science and empowering communities to monitor the health of their environment and their animals as well. So why? What is the need for this approach? Well, firstly, and I'm sure this is a conference where I'm preaching to the converted, but the Arctic is a place where One Health is very real. There are very strong links between animals, people, and the environment. So One Health is a very suitable approach, and in fact, probably the only approach that can work in the Canadian North, where you can't just come at things from one angle, can't just come at it from, for example, a wildlife conservation perspective. You also have to come at it from the perspective that wildlife conservation and wildlife health are food security and our human health in the Arctic, that they're just that intricately linked. And we can't just take a single viewpoint, nor can we just take a traditional scientific viewpoint. We also need to engage with the indigenous and traditional and local ways of knowing. And the other urgency for this network is that things are changing and that climate sensitive diseases are very likely to emerge in the North in a different future for the climate. And in fact, we're already seeing evidence that changes are occurring. And so that's where we're focusing most of our efforts from the infectious disease side, is on those diseases that are likely to emerge or change in distribution or prevalence or significance for wildlife, human health, and environmental health. So this is from a national study looking at food security in northern Canada, and it looks at 3 of our Inuit regions compared to the national baseline, which is the last bar here. And you can see here that in Nunavut, 70% of households, so the gray bar and the orange bar together, are experiencing moderate to severe food insecurity on a regular basis. That compares to 33 and 13% in the Nunavut region and roughly similar in Nunatsiavut. So all of the Inuit regions are experiencing food insecurity at much higher percentage than the national average, which is on par with 9% of households. So there's definitely a clear problem here, a clear need that food insecurity is rampant in the Canadian North. And these are some of the reasons why, and this isn't from me, this is from a food security coalition in Nunavut who recently came out with this list, but they point out that there's some demographic reasons why there are issues with food security in the North. But what I found very interesting was the last 3 reasons here. And one is that simply because of logistics, store-bought food is not always reliable, it's not always accessible, and it's also very, very expensive, particularly the healthier, fresher options. And so here you can see that for a 4-liter jug of milk that would cost me maybe $3 here in Saskatoon, it costs $20 in some of our communities. So not only is it expensive and not always available or a great quality by the time it gets there, but it's very expensive. The other unique aspect of food insecurity in our north is that changing access to hunting grounds and changing wildlife stocks, as well as concerns over the food safety of consuming wildlife, in part due to contaminants, and in part because wildlife health is coming under threat from climate change and other pressures. Those are also contributing factors to food insecurity in the north. And so here's a picture of empty shelves, which is not uncommon in some communities in our north. For the train didn't come in, the plane didn't come in. This is something that became more familiar to all of us during COVID, but for us it was a blip. For many residents of the north, this is unfortunately very common. And so instead of seeing empty shelves in the meat department of your local northern store, you may turn instead to the shelves of your community freezer, where you can see from this freezer in Nunavik, caribou are obviously a very, very important contributor to food security. And indeed, studies have shown that having a hunter in the household, having food, country food or harvested wildlife sharing networks, these are all ways that food insecurity is reduced in the Canadian North. And I just want to highlight that wildlife have a huge contribution to meeting sustainability goals in the Arctic. And One Health is actually under the Sustainable Development Working Group of Arctic Council. And wildlife contribute to all these things with all these squares with the black lines. So they contribute to alleviating poverty, alleviating hunger, to the health of populations, to education. Learning to hunt, learning to harvest wildlife is a key part of education. Wildlife contribute to good jobs and economic growth in the North, sustainable cities and communities, responsible consumption, protecting the planet, and of course, conservation of life below water and above water, as well as peace and justice, because it's food sovereignty and Indigenous rights to harvest their traditional foods are indeed a part of peace and justice. So wildlife is very, very important on many, many levels. And things are changing. The Canadian Arctic is warming at 3 times the global rate, which is pretty alarming and certainly makes the Western Canadian Arctic, where you can see is quite a hot spot, a real sentinel, a canary in the coal mine of climate change. So warning to the rest of the world about what climate change may do. So where our network comes in, these are 2 parasites that are transmitted through foodborne routes, as well as others for one of them, and they're the focus of some of my research. So Toxoplasma gondii is a single-celled protozoan. You can see here these are little muscle cells, and inside the muscle cell is a little tissue cyst full of little purple dots, and each of those would be one of the infective stages called bradyzoites of the tissue form of this parasite. And many, many mammals and birds can serve as intermediate hosts for this parasite, having these tissue cysts in their organs. But the ultimate source, the one that actually produces environmental stages of the parasite, are cats, domestic or wild. So the ultimate source is felids, but many, many other wildlife hosts and people can come in on the intermediate host stage. And there's many ways that this parasite transmits. Can transmit through foodborne, through water, through the environment, directly from cats, and also vertically for mammalian hosts infected for the first time in pregnancy can affect the fetus. And so this is a parasite that is extremely successful. It's all over the world. We estimate that 30% of the global human population are exposed, and in almost any system where you look, you can find Toxoplasma. The other parasite we study is Trichinella, and there's several different species. This one is solely foodborne, so it only transmits from carnivore to carnivore. And you can see here's a little tissue cyst. This is a roundworm, so it's a little bigger than the Toxoplasma cyst I showed you. This is a little curled up roundworm in the muscle cell of the host. And these guys get into the next host when it ingests the meat with these tissue cysts. It goes through a brief intestinal stage, and then they produce more of these larvae, which shower out through the circulatory system and end up in the organs and muscles of the next host. So it is solely foodborne. And both of these are important in the Arctic. Toxoplasma in particular, through something we call the Inuit Health Survey, which involves taking samples from communities across the Canadian Arctic, have found that there are high levels of Toxoplasma exposure, so antibodies, in people in Nunavut, so 30%, and in particular in Nunavik in northern Quebec. So as many as, some communities had as high as seroprevalence as 88% of people had antibodies to Toxoplasma. Overall, it came down to 60%. So in both those cases, it's substantially higher than what we would expect to find in the general North American population, which is on par with 10 to 15%. So obviously, in some regions of the Canadian Arctic, there is higher human seroprevalence. What's a little enigmatic about Toxoplasma is, of course, how it gets into the Canadian Arctic when the ultimate source are felids, or cats, which are not hugely common. Not, I mean, there are kept as pets in areas of our Arctic, but certainly not in the densities that we would see further south. And we certainly don't see a lot of free roaming domestic cats. We also don't see a lot of wild cats or lynx north of the boreal forest. So certainly there is a mechanism there for oocysts that are shed in the feces of lynx and other cats in subarctic regions to travel perhaps through runoff from the land into the ocean. So those oocysts could wash into the ocean or wash into freshwater and could be a source of exposure for Arctic wildlife and for Arctic peoples. The other way that Toxoplasma can get into the Arctic is through migratory wildlife. So it could hitch a ride in migratory birds who overwinter in southern regions and then breed in the Canadian Arctic. It could also come up with caribou who migrate to their breeding grounds, between their breeding grounds and wintering grounds. And of course, the marine mammals may be exposed in marine systems. So we think wildlife are a pretty important source of the parasite being introduced seasonally into the Arctic. So we know we have high human seroprevalence. There are some epidemiological studies to show that harvested wildlife are correlated with a higher risk of human exposure. And there's still outlying questions about whether or not Canadian lynx are indeed suitable definitive hosts. We know they have been seropositive, but any one of hundreds of mammals and birds can be seropositive for Toxoplasma. What has yet to be demonstrated is whether or not lynx actually shed the parasite in their feces. So that's something that my student Emilie Bouchard is working on. And then there's another twist to this in that foxes, Arctic foxes, who are at a fairly high trophic level, they're high-level carnivores and scavengers in the Arctic, appear to be pretty good sentinels for foodborne parasites and may have similar exposure routes and even prevalence as Arctic peoples. So some work by Emilie involved looking at quite a few red fox, mostly in Nunavik, and Arctic fox from across the Canadian Arctic, looking for Toxoplasma, either DNA in the brain or the heart, which are predilection sites for the parasite, or looking for antibodies. And in this case, we can't usually get sera from dead trapped foxes, but we can usually take heart fluid or in some cases filter papers, but we generally just use thawed heart juice and you can detect antibodies in the heart fluid. And so it shows that about a third of foxes appear to be infected and exposed to the parasite. And what was really interesting is that the prevalence in foxes really amazingly to us roughly tracked the noted prevalence in people, particularly in Nunavik. So it did seem like they were good sentinels for human exposure. And the other work that Emilie's been doing, and only Emilie can look so happy about a bag of digested tissue. The other interesting thing she's been doing is looking at stable isotope analysis of hair from the foxes to determine what their main prey source is. And you can see here that they sort out on their nitrogen-carbon ratios. The marine prey are up here, geese are kind of in the middle, and then the lemmings or rodents are down at the low end of this scale. And when we look at the Toxoplasma status of the foxes, we can see the foxes that ate more marine prey and geese were more likely to be seropositive. So that suggests that these marine sources, as well as geese, are pretty important sources of exposure for foxes and may also be for people as well. So sometimes we end up with a more practical question, and in 2014 there was quite a lot of excitement when Toxoplasma was detected in Arctic beluga in the eastern Beaufort Sea. So we came into the project after this finding to try and determine and answer this very basic question that harvesters had for us was, is muktuk safe to eat? And so we looked for Toxoplasma gondii in beluga whales that were harvested for other purposes. And over about a 4-year period, we looked at serology from about 100 animals. And when we first ran a modified agglutination test, we came up with a very startling result, which was that 96% of them were positive, which was a bit concerning. And one issue with agglutination tests is that they may They may have interference from high levels of lipid in the blood. Well, as you can imagine, Arctic mammals do tend to have high levels of lipid in their blood. So we did a cleanup step here, and you can see how much fat was in this one sample. But even the cleanup didn't really change our prevalence results that much. Using different tests, using an ELISA and an immunofluorescent antibody test, one animal was doubtful on ELISA, so kind of an equivocal result. That animal proved to be positive on IFAP. So that one we're pretty sure is a true positive. So antibodies were present. The other testing we were doing is the molecular testing using PCR to look at brain, heart, as well as spleen. And in this case, only one animal tested positive in brain. So this magnetic capture method that we use is really nice because for most PCRs, you can look at 25 milligrams of tissue, which is a very tiny amount. For the magnetic capture, you can actually look at 100 grams of tissue, and you use a capture method that is specifically going to pull the DNA of Toxoplasma out of a whole wash of host DNA and perhaps other pathogens and parasites as well. So it's supposed to be one of the better techniques for picking up Toxoplasma in foods. So the beluga that was positive on serology was negative on brain and heart, which is not impossible. It's quite possible to miss a tiny little microscopic tissue cyst in an entire animal. And the beluga that was positive on brain was negative on serology, which again could be simply that the animal was very acutely exposed and had yet to develop an antibody response, or perhaps it was a chronic infection and antibodies had waned. So of all those animals, about 2, we figure, were positive for Toxoplasma. So that let us get back to the communities with the message that although there were some positives, that we consider it to be a pretty low risk. For comparison, we see prevalences for Toxoplasma in Arctic fox and wolverine on par of 50%, 70%. So it's not, this is a pretty low seroprevalence, prevalence period. When we looked at other species, caribou are seropositive, but don't seem to be we don't find the DNA in the tissues very often. And ditto for ringed seal. We found seropositives, but not necessarily tissue positives. So those are still question marks in terms of risks for people. We did find that ptarmigan were not a source of infection for people, although we did find that geese were positive and probably acquired the parasite on their southern overwintering grounds and bring it up with them every year when they migrate up to the north. So the other parasite I work on is trichinella, that other foodborne parasite. And generally we see outbreaks of this in the Canadian North associated with consumption of bear, polar bear, grizzly, or black bear, as well as walrus. And there was a bit of a scare back in 2016 when there was an outbreak that was thought to be linked to seal meat in Nunavik. So there were some questions there. The other thing we know is that there is fairly high human seroprevalence for trichinella in Nunavut, and generally we would expect to see less than 1% in a human population, so a seroprevalence of 24% does indicate that there's quite a bit of ongoing exposure in the population. So the methods we use here, we take a tissue sample and basically we create an artificial digest where we recover the larvae from the tissues. And then we also use molecular methods to characterize them because there are different species of trichinella that can infect wildlife, including T2, the most common one, which is T. nativa, and T6, which is as yet unnamed. And you can see by the banding pattern on this PCR that you can pretty easily tell them apart. We also had a little interesting discovery here with T2 in that when we started doing deeper molecular characterization on isolates of T2 and T6. Instead, what we found was what we call a new old species of trichinella. So it was new to science, but it appears to be quite old, having branched off from ancestral trichinella a long, long time ago. So it's not something that evolved in the last few years. It's just been detected recently. And what was interesting about this and why we entitled the paper Hiding in Plain Sight was that on this PCR, our new species had exactly the same band as T2. So it was there but it just happened to have the same band as T2. And so this little cryptic parasite was hiding behind the molecular methods that are most commonly used. And when we looked at wolverine from across the Western Canadian Arctic, we found that it was distributed all the way up to the Alaska border and well into the Northwest Territories as well. And wolverine are great sentinels for trichinella. 80% prevalence is not uncommon, and a mean of 23 larvae per gram of muscle, which is fairly high intensity. And also many different species of trichinella were present in wolverine, including, and very interestingly, T. spiralis, which is the one we generally associate with pigs, and is supposed to have been eradicated from Canada. So we're still trying to figure out how it showed up right in the Klondike region near the Alaska border in a wolverine. So some of our work on trichinella has concluded that we did not find it in the ringed seal that we've looked at so far. We did not find it in beluga so far, and so those are unlikely to be common sources of human infection. And we are also interested with the idea of using dogs as sentinels for trichinella if they're often fed raw or undercooked meat. And this was a really unique study that we got pulled into, looking at a long-term data set, sera from polar bears in the Western Canadian Arctic. So here is the home range or study area of this population of polar bear in Western Hudson Bay. And we looked at samples collected in 3 different time periods, one in the late 80s, one in the late 90s, and one in 2015, 2017. And we looked at a range of different pathogens, but for the 2 I just discussed, Toxoplasma and Trichinella, we saw seroprevalence increase for Toxoplasma over the 3 periods, and not quite as clear a pattern in Trichinella. And what was very interesting is that we looked at climate conditions during this time periods. And Toxoplasma had higher seroprevalence in years that had wetter summers, suggesting there might be a oocyst transmission through water component. Trichinella had higher abundance or seroprevalence in years when we had warmer temperatures. So that might be that the carcasses stay thawed longer, or that the larvae, which are freeze tolerant, might survive longer in slightly warmer conditions and therefore be able to complete their life cycle. And then we also looked at a bacteria called Francisella tularensis, the cause of tularemia. And there we found that it too had a upward trend. So over the last 40 years, it has increased in seroprevalence, and it was correlated with the years that there was less sea ice and where polar bears spent more time on land, suggesting that this is a pathogen that is largely exposed, that largely circulates in the terrestrial ecosystem in the Arctic. And all of these are zoonotic. People who eat polar bear that's undercooked could be exposed to Toxoplasma or Trichinella. Not so much tularemia. We think they're probably more of a sentinel for tularemia than a source of human infection. Nonetheless, the links here in this long-term data set that show a steady increase over the last few years, as well as strong links to climate, really gives us a picture of what might be happening already in the Arctic as a result of climate change. So vectors and vector-borne diseases are the other main aspect of this work. And tularemia is also a vector-borne disease. It can be transmitted through a range of insects in the Arctic and elsewhere. But we more broadly are looking at vector-borne diseases, including Bartonella and other bacteria, as well as a group of viruses called the California serogroups, and a nematode of caribou called Ceteria. And everybody knows there's lots of mosquitoes in the Arctic, but we actually don't know all that much about the diversity of mosquitoes. And so that's another aspect, is looking at just what arthropods are present, and is that changing or likely to change as climate changes in the next few years. So for Bartonella, which is in the cause of cat scratch fever, so here we have Toxoplasma, a cat parasite showing up in the Arctic at high prevalence, and now we have this cat-borne bacteria, which is a bit misnomer because it does seem to be present in a wide range of wildlife. was isolated from 3 Arctic foxes at Carrick Lake, Nunavut, which was very interesting because, again, no cats. And it's also transmitted by fleas, which Arctic foxes don't have a lot of their own fleas, at least that we've ever seen. However, they are based in a large goose colony, which has many, many, many nest fleas. So there was some possibility there that the geese fleas were actually transmitting this parasite to the foxes. So we decided to explore that hypothesis. The hypothesis being that geese infected with Bartonella would come up seasonally, that they would then transmit it to the nest fleas, who could then bite Arctic foxes when they're harvesting the eggs or eating the chicks. And so that there was probably this seasonal emergence of Bartonella every year brought up from the south, as well as the potential for endemic Bartonella species circulating between rodents, which are known normal wildlife reservoirs, for a wide range of species. So we thought it would be relatively simple. And then we started looking more deeply into the species of Bartonella and found that there wasn't just Bartonella henselae. There's also this Bartonella vinsoni, which is more associated with canids, Bartonella rochellimi, which is associated with rodents. And so this picture rapidly became a lot more complicated than we thought. And so this was a nice case study also showing the importance of migratory birds in bringing pathogens seasonally into the Arctic. So on the mosquito side, some of the work that our students have been doing, and I encourage you to look at their posters, Caroline Villeneuve, has found that there are species, we detected species in the Arctic which were not previously considered Arctic. So again, they could have just been missed in previous biodiversity scans, or they could be genuinely moving north. And the other thing we found in the mosquitoes is evidence that they were transmitting these California serogroup viruses. In Nunavik and Nunavut, we found evidence of Jamestown Canyon virus, which is associated with cervids. And in Nunavik, we also found snowshoe hare virus, which is more associated with rodents and rabbits. And finally, we also found Jamestown Canyon virus in biting flies in Nunavik, so not just mosquitoes. So following up a little bit more on the wildlife reservoirs for Jamestown Canyon virus, we knew that in southern Canada and in the US that deer are a natural reservoir. There was a really interesting study that came out recently in Alaska that showed a human seroprevalence of 27%, so almost a third of people in Alaska were positive for exposure to this virus, which doesn't generally cause severe disease. Rarely it can be associated with neurological signs. But this suggests that there is endemic levels of transmission in the Arctic. And in fact, we had the opportunity to sample captive reindeer out at the Large Animal Research Facility near UAF. And it was really interesting. We sampled a cohort of yearlings in May of 2020. So they'd had one summer of being outside, and only 2 of them were positive. And then when we resampled them again after that summer, almost all of them were positive. So there definitely is high levels of natural exposure to this virus circulating in Alaska. And so our goal was, and this is again, see poster by Kayla Bueller, goal was to figure out if caribou or reindeer are good reservoirs and if it has any effect on their health. And so we experimentally exposed captive reindeer, some who were naive and some who had a previously low titer. And we did see a seroconversion at 5 days, elevated eosinophils and lymphocytes, and we did detect virus in blood, brain, and other organs. So it suggests that caribou are viremic, that they have the virus in their blood, so they are a source of infection for the mosquitoes, but that they don't seem to get severe clinical disease associated with it, which makes them ideal reservoirs. And indeed, when Kayla was sampling free-ranging caribou across the Canadian Arctic, we saw fairly high levels in the Western Arctic, up to 83% in some regions, and a lower level in Nunavik. So tularemia, I mentioned previously as well, We've also looked at other sentinels for tularemia, and again, Arctic foxes seem to be great for picking up everything. So they could get this bacterium either from rodents or from vectors. And thus far, we've tested rodents and have not found tularemia. But the irony here is that it can be quite lethal in rodents and rabbits, and so it might just be that we don't find positive animals because they die. And we haven't found it yet in Arctic mosquitoes, but again, there's a lot of mosquitoes, so there could just be a dilution effect there. The one interesting thing we have found in a multi-year study in a mark recapture population of foxes is that there's this evidence of a tenuous link between vole numbers. So when vole numbers are high, the next year we saw very high seroprevalence of tularemia in foxes. And indeed, this fits with data from Finland, where they see human cases peak the year after a high vole abundance year, which is of course quite cyclical. So again, foxes might be very good sentinels, showing that there's high levels of exposure probably through rodents. And this was a really interesting finding that was first noted in 2019, again in this fox population that has been the subject of a 20-year mark recapture project with Environment Canada. And for the first time ever, we saw foxes with this bizarre fur loss over their dorsum. So this is atypical of how they would normally shed their coats in spring, which starts more on their posterior. And on close examination, we found a few nits or little eggs associated with hair of these affected foxes. So this was interesting. We have a new website that our grad students put together tentatively called the Northern Animal Health Alliance, which is a way for communities to know what research is going on in their area if they want to get involved. They know they can find out the researchers and they can get results straight from the horse's mouth instead of having to read scientific papers. So far it's still at the beta stage. We're trying to roll it out, getting some feedback as we go along. But here's an example of highlighting some of the Arctic fox work and who you would contact. And we also have a blog, which is where we wrote a little article talking about how this new fur loss syndrome, which could be associated with lice, is something that would be very well suited to community-based monitoring, because fox trappers might see this and we can start looking at, is it something that happens when we have high fox abundance? Is it linked to climate? So yeah, it's definitely something that would lend itself well to the two-eyed approach to wildlife health surveillance. So our future directions for our network, well, we would like to expand it geographically. So not just the Canadian Arctic One Health, but maybe a circumpolar Arctic One Health network. And remember to go beyond just zoonoses, because although those are important at the interface of animal and human health, there's a whole lot more to One Health than just zoonoses. We'd also like to follow up on that new species of trichinella to see if it is distributed in Alaska and or Eastern Siberia, if it's something that came across the Bering Strait. Other aspects of this work include wildlife disease two-eyed viewpoints, which are well described by Susan Kutz's talk. Also One Health governance, which isn't just government and politics, it's looking at bringing together stakeholders needed to address complex problems at the One Health interface. And finally, we're also starting to do some monitoring for COVID spillover into Arctic wildlife and Arctic environments. As this pandemic becomes more managed in people, we are more concerned about its ability to establish in wildlife reservoirs. So many different partners involved in this work. Without the northern partners, it wouldn't happen. So they're absolutely fundamental, but we work with almost all federal government agencies, CFIA, Environment Canada, Fisheries and Oceans, Public Health Agency, Polar Knowledge, as well as NGOs like the Canadian Wildlife Health Cooperative, Veterinarians Without Borders. And this was our group, my personal group, pre-COVID, and this is how we look nowadays. So thanks very much. I'd be happy to answer any questions.
Hi, I'm Dr. Ann Zink. I'm the Chief Medical Officer for the state of Alaska, and it's great to be here with you today at the One Health Conference and talking a little bit about COVID-19. It's great to hear from the other panelists, and I just wanted to share a few slides on where we're at with COVID-19 and talk about a few other topics along those same lines. So going on to the next slide, thinking about zoonotic diseases in general, COVID fits within that. As many of you know, COVID is an enveloped RNA virus that circulates in animal hosts and can move to human hosts and also circulates in human hosts overall. As you all know, coronavirus is named for its crown-like spikes on the surface that we've all gotten to know well, particularly with vaccines. And they have different subgroups within them. There's hundreds of coronaviruses out there, most of them circulating amongst animals such as pigs, camels, bats, cats, and so forth. But there are 7 coronaviruses that infect people, including ones that cause the common cold. So about 10 to 30% of common colds are from coronaviruses that kind of circulate amongst the population. But moving on to the next slide, there have been a series of more recent introductions of coronaviruses, likely from zoonotic origin, including what we know of as SARS, caused by the virus SARS-CoV or SARS-CoV-1, and then more recently MERS, or the Middle Eastern Respiratory Syncytial Virus, and now most recently COVID-19 caused by the virus SARS-CoV-2. All 3 of which have circulated amongst humans. SARS, as people know, no longer circulating, but MERS and COVID continue to circulate overall. Moving on to the next slide, just a little bit of a comparison between these viruses as well as a couple other viruses, questions we get asked all the time, and really looking at the case fatality rate, again, different than the mortality rate, and this changes over time as we learn to deal with diseases differently. But SARS in general has a case fatality rate of about 10%, while MERS was much higher, about 45% case fatality rate. And as mentioned, it continues to have animal reservoirs and spill out intermittently with sporadic zoonotic spillover. Influenza H7N9 or bird flu, really high case fatality rate, about 56% there. Influenza in general, case fatality rate on average about 0.1%. H1N1 even lower, very wide estimates, but maybe somewhere around 0.03% case fatality rate. And then thinking about other viruses like Ebola, we're getting up to more like 60% case fatality rate. And then COVID-19 kind of fits perfectly within there. It doesn't have such a high case fatality rate that you can identify cases early, and such a high asymptomatic spread that it can quickly move from person to person. And one of the just really challenging things with messaging is how mildly it can affect people and then how severely it can affect people. And it can be very hard to predict who is going to be that person. We've seen wide ranges of estimates of the case fatality rate over time looking at COVID-19 as we've learned more about the disease, been able to identify it earlier, and as we've gotten better treatment options, including things like monoclonal antibodies, steroids, being able to watch people's oxygen. But this is the most recent data I could find from Johns Hopkins, and here we see case fatality rates in the US ranging about 1.8%. When we look at places like Mexico, we're getting higher, about 9.1%. And in general, the case fatality rate has come down over time, but still much higher than influenza or H1N1, but much, much lower than SARS, MERS, or Ebola. But because it can spread so easily, that's part of the reason we see such high mortality. Just so many more people are infected, resulting in more deaths overall. So you all have been a part of this journey with us with the COVID-19 response here on slide 6. And there's just been so much that has been learned. I saw a fun presentation the other day, COVID-19 by the numbers. And one of my favorite numbers has been, 63 days from the time this virus was first sequenced to the first vaccine was trialed in a human subject. So it's just been remarkable to watch the science on this, from first identifying this kind of unknown pathogen to sequencing it, sharing it worldwide, and communally working as a scientific community to learn about this disease. I remember learning about it in late December, and man, things have changed in that short period of time. And here we are on slide 7 with being able to get a remarkably effective vaccine out across the state of Alaska into all sorts of wild and wondrous places to be able to protect people against the virus. It's impressive to me how many people are really still so concerned about the side effects compared to the benefit and don't see the full effects of COVID-19. And so I think it's really important when you're talking to loved ones and friends and community members to help to put these numbers into perspective and what COVID really can do to an individual versus how remarkably both safe and efficacious this vaccine is over time. And this is, we don't have good ways to treat viruses in general besides vaccines. This is our way to prevent against the virus and be able to teach our immune system to move forward. So really doing everything we can to encourage people to really understand these numbers of how effective the vaccines and looking forward. Moving on to the next slide, talking a little bit more about antibiotic resistance and what a big role that this plays in our overall ability to deliver healthcare. And I think there's a lot of similarities in some ways between COVID and antibiotic resistance and different limitations in the data. Overall, each year in the U.S., there's at least 2.8 million people who get antibiotic-resistant infections, and more than 35,000 people die every year. And this continues to be a growing public health threat across the nation and across the world. We've really had the last 100 years of remarkable health with vaccines as well as antibiotics. But we can see with viruses that we're starting to get more and more kind of zoonotic spillover. And then we are also starting to see more and more antibiotic resistance. And I think these will be huge public health challenges moving forward. In a way to try to reduce antibiotic resistance, we do have the antibiotic stewardship programs, which can be really fundamentally important to helping clinicians improve clinical outcomes and minimize the harm by really improving antibiotic prescription. The programs can increase infection cure rates while reducing inappropriate prescribing, treatment failures, adverse events, antibiotic resistance, and reducing hospital costs and length of stay. But kind of like COVID response, it requires a really multidisciplinary team to be able to get the right place and the right tools moving forward. Here's a study on page 10, a Pew study looking at antibiotics with COVID-19 specifically. And you can see that 52% of people received at least one antibiotic during admission for COVID, again, a viral infection. 36% of people received multiple antibiotics during their admission. But it can be just so hard to tell on the very front line. You've got someone who's hypotensive and their heart rate's high and they look really sick. And is this a secondary bacterial infection? Is this primarily just COVID? And really with these septic measures that we have to perform at now as a nation, it can be really challenging, and you are significantly dinged if you do not give the right amount of fluids and antibiotics within that first hour. And we see with sepsis real improvement with getting antibiotics on early if you suspect a bacterial infection, but clearly they don't do anything for a viral infection to the same degree. And so really being able to kind of help support clinicians in that real-time decision-making on choosing antibiotics or not can be a really important tool moving forward. In fighting antimicrobial resistance and COVID-19, here on slide 11, we do have the Alaska Antimicrobial Stewardship Collaborative, which has been a great partnership really with working with clinicians, scientists, being able to look at the data to provide real-time tools and including state funding to health departments to really have improved antibiotic stewardship for making the decisions locally based on antibiotic resistance. Moving forward and thinking a little bit about where do we take everything that we've learned into the future, I feel like how we actually responded to this pandemic, it was important, but how we recover out of it is going to be even more important moving forward. So what do we do in terms of emergency preparedness? How do we respond to the next pandemic? What are the lessons learned and what can we do moving forward? I think for Alaska, it's always important to put Alaska in context. We're larger than the 3 largest states combined, 229 remote villages, many accessed by plane. We see different antibiotic resistance patterns in different areas. We see TB, we see tetanus, we see rabies in different regions of the state. And so understanding those patterns can be really important for figuring out the best treatment options as well as what sort of surveillance we need to do. On to the next slide, being able to visualize and get real-time data out to communities, making sure that it is bidirectional and actionable is incredibly important. We spent a lot of time trying to improve these data systems, but we have a long way to go and really need all Alaskans' partnership in this process, making sure that we've got good syndromic surveillance data from hospitals, making sure that testing results are being able to get in a timely manner, including information like gender, age, and race and ethnicity that can help us to be able to respond better, and making sure clinicians on the front line have access to that data on the front line so they can make good decisions about the antibiotics they use. Is this really COVID or is this something else that's happening? And being able to make those clinical decisions in real time. So as many people know, we've stood up the Alaska Coronavirus Dashboard Hub, and our plan is to continue to just really work on how can we visualize data in real time. We've been doing this for flu for a long time with our weekly snapshot that looks at kind of flu patterns across the state and continue to track that, but continuing to look at how we can build and take these tools that we've used together. Going on to the next slide, I think testing has also been really interesting in thinking about our response. We really made sure that it was accessible, affordable, and Alaskans could get it when they needed it. And so thinking about what ways kind of outside of their traditional healthcare infrastructure can be built in ways to support people. Again, not having systems, people serve systems, but systems serve people, making sure that people are able to get the information and tools they need to promote their health, be that, telehealth for behavioral health, be that quick and rapid testing to be able to identify a disease process that may change your behavior, such as a positive COVID test. I think another lesson that's really been learned has been communication and partnership with our partners. This is early on in the pandemic with Mayor Morris out from Stebbins, and really hearing what that looks like on the ground. If you have a community that doesn't have running water and sewer, it's hard to say, wash your hands. And so what are the alternative ways to being able to message? How can you really empower communities to change their future of their health? Stebbins was a great example when they had an Abbott Now machine, was able to really kind of keep an outbreak under control very easily by having the tools and resources that they needed to do it before it really overwhelmed their community. And now thinking about what ways we can promote the overall health and well-being, what ways can we build our vaccine process, not just having a children's vaccine process, but also having an adult vaccine team. Currently, we've built a lot of infrastructure along those lines, and everything that we build in our COVID-19 vaccine team, we're very much thinking about how we can make this long-term, how we can make this sustainable, and how we can make sure that vaccines are easy and accessible across all age groups for all sorts of different diseases that we do have vaccines available for. And I'm honestly really excited to see the technology and what I think is going to just be a real growth in vaccine development because there's been so much science that has grown out of this last year and being able to respond to the pandemic. Again, in an effort for transparency, you can see our dashboard overall. So you can look at, 65 and plus, we're almost at 72% of Alaskans have received at least one dose. If we look at Alaskans 16 and older, we're at 40.6%, which is just fantastic to see. So we're really excited to see those numbers. We have really flattened out. Our slope is not what it used to be. And so just really encouraging, particularly providers, to share their experience. Over 90% of healthcare providers have received vaccination. And I think that really speaks to the healthcare community's trust in the vaccine, as well as the fact that they've seen what COVID can do. And the side effects are so minimal compared to the really severe outcomes that we really do see with COVID-19. And so thinking about kind of the three pillars of what we learned, I think there's kind of three major things I've been thinking about a lot. One is communication, really that partnership led by communities, making sure that data is real-time, transparent, it's visualized and meaningful and actionable. And it's two-way dialogue that really increases trust and strengthens response, and making sure there's places for that two-way dialogue like our echoes. Building data results in better responses. So convenient, low to no cost testing outside of traditional healthcare, uniform reporting metrics and bidirectional information is key for action, and reporting that's meaningful and actionable, not just having reports for report's sakes, but creating data that we're able to action so that we have a good sense of what's happening with the health and well-being of our communities so we can smartly invest resources where we need them. And again, at the end of the day, putting patients first. I think one of the things I love the most about DHSS is our mission, and that's the health and well-being of Alaskans. And man, that is an easy place to stand behind, and always having that goal ahead. It's not to reduce our COVID numbers. It's not to reduce anyone numbers. It's to build the health and well-being of Alaskans. One of the challenges to that has been this virus, but we also need to be thinking about the mental health, the overall physical health, making sure people are getting their regular childhood immunizations, the rest of their regular preventative care, and making sure that that whole person is taken care of moving forest. So I just really want to thank Alaskans for their partnership and remaining prepared, investing in public health, and continuing to grow in these partnerships. And really want to thank particularly Alaska Native people for the history of the 1918 pandemic. I think that's really shaped our response. And then I think thinking about how we sit in this kind of larger perspective of our world and of our nation, thinking about the interface between viruses and bacteria and human in a larger global scale is going to be really important in thinking about how we respond beforehand. This woman here, when she was getting her vaccine, says she's getting protected from the great sickness, but she had all sorts of great stories about the great death from the 1918 pandemic. So lots to learn and lots to move forward on. So again, thank you for your time and partnership, and looking forward to the rest of the conversation.
Hi, this is Peter Rabinowitz, and I am a physician and a faculty member at the University of Washington, where we have a Center for One Health research. And it is a real pleasure to be part of this One Health conference from the University of Alaska Fairbanks Center for One Health research. And I'm going to talk today about a zoonotic disease. Since this is a zoonotic disease section, I'm going to talk about bird flu, and especially avian influenza in a changing Arctic climate. And I want to go back to a couple of years to a major outbreak of bird flu in the United States. So some people think that bird flu is all something that happens over in Asia, but we had a major outbreak in 2014 and 2015, and it was actually the largest animal health disaster in the history of the United States, and up to 50 million birds died. It was sort of started right around the winter into the spring. It affected chickens and turkeys and all over both the Pacific part of the United States and the Midwest with this avian influenza strain. Fortunately, no people got infected or died, but a very slight difference in the strain could have made a difference in that. And when people started looking at where these outbreaks were happening, they saw that it was really first introduced in Washington State. In terms of the continental United States, that's where it was first detected after being in British Columbia before that. And then it rapidly spread down the Pacific, but also then in the Midwest as well. And it was mostly in backyard and commercial poultry flocks. But you can see the yellow bars here, the squares, are where wild birds were also detected. So there was a connection between wild birds and this poultry outbreak. And the Department of Agriculture realized that the reason it was in the Pacific and the Midwest was that these are where there are major wild bird flyways, because wild birds spread influenza. The wild waterfowl are the reservoirs for influenza. And they started thinking about the flyways, and that really brought attention to Alaska and Beringia, where it was recognized that the highly pathogenic bird flu, which was circulating in Asia, had gone up through the East Asia-Australia flyway into Alaska, and then birds from Asia were mixing with birds from North America that then moved down in the Pacific and Midwest flyways. And so Alaska was really playing a central role in the mixing of influenza in birds from Asia and basically the mixing with the United States. And so this is just sort of showing these overlapping flyways that birds all the way from down in Australia will migrate all the way up through Asia into Alaska, and a lot of movement from different parts of Asia, and then movements in the Americas all the way down to South America as well. So the real central point is Alaska in all of this. And what can happen is locally, if there is birds getting together, we know from our experience with Asia that migrating birds in contact with domestic animals can lead to outbreaks in domestic birds, and that can be an outbreak in a market, or if you're a subsistence hunter, it could be direct exposure to an infected bird. So just remembering about what influenza A is that it's really a waterfowl disease. It really has its worldwide reservoir in ducks and geese. And occasionally when there are these reassortment of genes between avian influenza and human strains, you get these pandemics. And we've seen that as recently as 2009, when both a wild bird strain and a swine strain came together with a human strain to create a new pandemic strain. So we think a lot of this comes from Asia as well, but remember that really globally it's the ducks and geese that are the main reservoir, although shorebirds and gulls can also play a role in infecting other animals that are not reservoirs but get sick, like people and pigs and horses, etc. So this is just a diagram that shows a number of things going on to tell this whole story. We've got basically the ducks moving around between Asia and Alaska and the United States. We've got the birds basically moving the virus down into the United States. We've got in the wintering grounds for the birds, we've got potential for local transmission from wild birds to poultry, and then we've got a number of places for entry into human populations. So in Asia, you can have farmers or market workers directly contacting either wild birds or domestic birds and getting infected there. Up in the Arctic, think about people who are having subsistence lifestyles and are eating waterfowl or shorebirds. They are directly exposed to birds that could be carrying viruses. And people working on small farms versus large farms are other sources of entry into humans where it could cause a influenza outbreak in the humans. And so much of this is related to what's happening in the environment where ducks and geese are mixing up in Alaska. How are they exchanging the virus between them? And what is the role of warming temperatures that are happening in the Arctic in the terms of this very important mixing and transmission in the environment of bird flu going on in Alaska? We are trying to get a handle on that. We don't really know. So we've been sampling animals and birds in Alaska for bird flu. There was a lot of money for it in the late 2000s because of major bird flu epidemics around the world. There was a lot of pandemic flu preparedness money, and then that all dried up and people stopped testing. So up until recently, we really don't know what's been going on in the wild birds. We did see a lot of virus in both pintails and mallard ducks when we were looking, but now we've just stopped sampling. There is money now coming with some new global health security money. It looks like there's going to be more sampling this year in Alaska, so we're going to get a picture again. But over time, it's been a little unclear, except that there's a lot of different species involved. And then it's also unclear in the environment how this is going to really play out in terms of a warming global temperatures and a warming Arctic environment, and is that going to affect in the water some of the transmission? Because we think that the birds poop in the water and could spread the virus from one animal to another, from one bird to another. And we know that as the temperature gets colder in the water, the virus can live it a little longer. And as the salinity goes down, so fresh water, there's more persistence of virus than in salt water. And when the water becomes more acid, that lowers the persistence of the virus. So depending on what happens to the water bodies in Alaska with climate change, you could get either more or less transmission of the virus going on. But we think that there's a real good chance that it could lead to more transmission. And one of the other things with climate impact is what's going on in both Asia as well as Alaska, but in Asia, what's happening with the land use in terms of the amount of farming and how that could potentially change the interactions between wild birds and domestic birds. If you have a lot of rice paddies and waterfowl everywhere right near chicken flocks, could that change the evolution in Asia? And then how could that relate to the risk when we get to places like Alaska? And we know that climate change is going to be more marked and severe in Beringia than many other places, that these higher latitudes are going to have more warming with melting of the permafrost. The water temperature is going to get warmer, but there's going to be changes in the water chemistry that, again, could either help or hurt the chances of transmission of something like avian influenza. So it's going to be really important to monitor this very closely, and as things start becoming clearer, doing something about it to mitigate the risks. This is a picture, by the way, of the kind of more recent temperature zones in the Arctic, with red being hotter, and this is just what's going to happen in the next several decades with increasing global warming. It's going to be very pronounced in Beringia. And what we've seen already with ornithologists who've been studying the birds is that the warmer it gets, the longer birds will be up in Alaska, which kind of makes sense. The longer the sort of the breeding season there, with some warming, so that they see in very cold areas of Alaska right now where there's a very short breeding season, the birds just aren't there as long. So these circles relate to how many months there are a lot of birds being seen. And you can see that where it's a little bit warmer, there's birds kind of over a very long period of time, compared to up north around Bethel or something where there's just very short period of birds there and mixing. So that warmer temperatures, you'd think, would give more chance for more bird interaction between birds from the Americas and birds from Asia. So, what does the future mean for avian influenza, for zoonotic diseases in general, but avian influenza in particular, with the coming climate change and other changes in Beringia? Well, we know that the climate could have a couple different avenues. It could change the avian migration routes and the timing. It could also affect agriculture and hunting practices and the water characteristics. So there's a lot of uncertainty. Will you get more transmission or less transmission with the warming? What are hotspots? Are there particular areas of Alaska or the region where we should be really looking for this to be happening more? How is climate change going to really affect the bird behavior, and how is it going to affect changing agriculture? How is that then going to lead to changes in influenza? Because flu has been around for a very long time. It's still as bad as COVID-19 is. The 1918 flu pandemic was worse, and just shows that flu is capable of coming back periodically in a very severe way and having a huge disease impact. And what's going to be the impact on populations all over the United States, including Alaska, but especially what about the indigenous populations that are subsistence hunting animals, and what's going to be their exposure, and what should we be doing about that? And it all argues for a One Health approach. This is a One Health problem that really needs a One Health approach. If we're doing surveillance to try to understand the situation and how it's changing, we should be doing surveillance not just of birds. We should be looking at humans like hunters and farm workers, and we should be looking at the environment and not just the birds. We should be looking at the water temperature, the virus in the water, salinity, pH, all those kind of things to try to understand what is happening in terms of are we setting ourselves up for more of this kind of disease problem. So we should be modeling and monitoring. We should look at hotspots. We should look at trends over time. We should look at the timing of the migration and the bird behavior and the bird migration, and use the bird sightings and understanding how the birds are reacting to all this as well. And this should all lead to interventions. We should be able to figure out how risk situations are developing and be educating and anybody who's potentially being exposed, find ways to reduce their risk, but get early warning of outbreaks. And if we can obviously do anything else in terms of environmental mitigation, we should be trying to do that as well. So it's a serious problem. It's a One Health problem, and it demands a One Health approach. And really look forward to more discussion about this. So thanks a lot. We're looking forward to more contact between the Alaska One Health Center and our One Health Center, and really look forward to collaboration on a number of different types of projects related to these issues. Thanks a lot.
Well, hi, everyone, and thanks for coming to this presentation. My name's Tom Hennessy. I'm with the University of Alaska, and I'm going to tell you about the results of a workshop to prioritize zoonotic diseases in Alaska. The workshop was held 2 years ago in Fairbanks, just prior to the previous One Health conference that the University of Alaska Fairbanks put on. And we were really seeking to enhance our One Health capabilities in Alaska by prioritizing zoonotic diseases to help us with research, response, and coordination. So you might wonder why a group would go about creating a prioritized zoonotic disease list for a jurisdiction like the state of Alaska. But we saw a need to focus our activities. Funding and personnel resources are limited, and we thought that if we could focus in on key areas together, we'd make more progress. Also, laboratory tests and equipment can't cover all diseases, so there may be a need to focus in on certain key priority diseases. We also saw a need to coordinate. Surveillance is better if it's coordinated across sectors. Many groups, despite working in One Health, continue to work in silos within their organizations and with their own priorities, sometimes with little connection to others. And we saw this workshop as an opportunity to build bridges across organizations. And we thought that a prioritized list could help with collaborative efforts where groups could add different kinds of expertise to solve problems around zoonotic diseases. And then we thought that because there's a need to get funding support, that a list of priority diseases is more likely to interest funding agencies. And we had seen that with several announcements with US government agencies where they wanted to fund groups that were dealing with priority problems for states, and having a priority list for Alaska could help with grant applications or other kinds of support. We were aware of the US Centers for Disease Control and Prevention's One Health Zoonotic Disease Prioritization Tool. They published the methodology in PLOS Science, shown here in 2014, and the URL link is below, and basically come up with a methodology to use a One Health approach to prioritize endemic and emerging zoonoses. It was flexible and scalable. It allowed for local adaptation, and even allowed you to prioritize conditions in the absence of reliable data, say on prevalence. And by producing a timely outcome, you could maximize the impact. And then the list could be used to focus financial and personnel resources in ways that we'll get into. So we were aware of this tool, and we are aware also that the CDC One Health Office had conducted workshops using this tool in a variety of locations around the world, mainly in Africa, but also in Asia. And we asked them to help us in Alaska set up a zoonotic disease prioritization workshop. Here's an example of one of their workshop reports from Uganda. They published these, put them up on the CDC website, and then the country has this tool that they can use moving forward for planning and action. CDC also published a review of the first 7 of these workshops that they held. This was in a screenshot from Emerging Infectious Diseases, and the URL for the article is there, down there below if you're interested in reading more about it. So we took our cues from the Centers for Disease Control and Prevention, got them to partner with us in organizing this One Health conference, and held it. So just want to walk through some of the actions that we took and what we came up with. So the goal of our workshop was to use a multi-sectoral One Health approach to prioritize endemic and emerging zoonotic diseases that are of greatest concern in Alaska, and that should be jointly addressed by sectors responsible for human, animal, and environmental health. It's a mouthful, but it is a consensus plan and goal that we came up with for the group. The workshop process is basically facilitated group work, meaning that there are group leaders that are outside to help move the process along, and we bring in One Health stakeholders that include state, national, and local government, indigenous groups, university groups, groups from human and animal, veterinary medicine, public health. There are also user groups of the resources, researchers, and land managers. We then use a structured decision-making approach that includes qualitative and quantitative methods, and we come up with a consensus list that's the final result. So this graphic I'll return to a couple times, but it is an overview of the 5-step prioritization process, starting in the upper left-hand corner, from preparation to facilitated group work to the final prioritized list. So in preparation, we had a group that developed the outlines for the workshop and did some of the groundwork for invitations and getting the setup, and began about 3 months prior to that. We came up with a list of 35 zoonotic diseases for the group to consider in prioritization. These included bacteria, viruses, parasites, prions, fungi, and then other diseases, which we included paralytic shellfish poisoning as others to consider. Although not technically a zoonotic disease, it really does act like a zoonoses and is a particular concern in Alaska, so we included that on our list. Most of these conditions are reportable to the state of Alaska, either to the veterinary or the human public health side of that. We then sought participating organizations who could bring people to the workshop. This included groups from the state of Alaska, indigenous groups, local government agencies, the University of Alaska, and US federal agencies all listed here. We had about 50 people that were invited and about 35 people showed up for the 2-day workshop. Day one of the workshop included the real start of the facilitated group work and included 2 main steps. One was to develop the criteria for assessing these 35 diseases. And the second step was to develop questions that we could apply to each of them to help prioritize them. And let me show you how this worked. So the five criteria that the group came up with during the day one of the workshop included these: the clinical outcome, the prevalence and modes of transmission of the disease, the food safety, security, social, and economic effects of the disease, our state's response capacity related to the disease, and whether the disease was going to be affected by climate change. We then came up with a series of 5 questions for each of these 5 criteria that we could develop objective criteria for and answer for the scoring process. And here are the 5 questions for each of the conditions. For clinical outcome, we asked whether it produced mild or severe disease in humans, in animals, or both. For the prevalence and modes of transmission, we wanted to know if it had occurred in Alaska and if it had one mode or many modes of transmission within our state. For the food safety issues, we wanted to know if the disease affected food security, food safety, local culture, or the economy. For response capacity, we were really interested in those conditions that we did not have response capacity for example, that were not well developed. So we asked how many capacities are not available within our state. And these included surveillance, treatment or vaccination in humans or animals, and outbreak response capacity within the state. And for climate change, we wanted to ask whether it was going to be affected or likely would be affected by climate change in the next 10 years, either increasing or decreasing. So then what we did when we had those criteria and questions, we ranked each of the criteria using a semi-quantitative method that used a hierarchical process, and we had the workshop participants fill out a questionnaire and then scored those questionnaires to come up with a scoring mechanism. So this is our ranked prioritization. Each of the numbers after each of the criteria add up to a level of 1, and basically the scores for these became multipliers for the criteria that we came up with, and this is how we came up with the objective part of it. So clinical outcome was ranked the most important with 0.36 weighting, followed by prevalence and modes of transmission, 0.25, food safety, security, etc., 0.21, response capacity, 0.1, and climate change, 0.08. Then, on day 2 of the workshop, we worked on the zoonoses ranking. Between days 1 and 2, the organizing team did a bunch of data entry on those criteria that I had listed in the previous slide and entered the data into a spreadsheet format that we could then run this prioritization on and answer each of the questions so that each of the diseases came up with a score relative to the other ones. This scoring then was shared with the group on day 2 for them to evaluate and to look at the process. This is really important because we didn't want to just let the computer decide. We wanted to bring the ranked zoonotic disease listing back to the group for the group to work through in a dynamic process and to see if there were any adjustments were needed or if we needed to rerun any of the data. And this is what we came up with after our consensus discussion on day 2. These are the top 7 zoonotic diseases of greatest concern in Alaska for One Health collaboration. Number 1, amnesic shellfish poisoning and paralytic shellfish poisoning. Number 2, zoonotic influenza. Number 3, rabies. Number 4, cryptosporidiosis and giardiasis. Number 5, toxoplasmosis. Number 6, brucellosis. Number 7, Q fever. I think it's interesting to compare the Alaska list with the United States list for prioritization through a workshop that had been done on a national level in our country. CDC sponsored this, held national stakeholders to begin, and you can see the comparison of the 2 lists. I've underlined the 3 conditions that appeared on both of our lists, but you can see the big differences from Alaska to the United States list. Number 1 on the US list was zoonotic influenza. This workshop was done in 2017, just a few years after we'd had a worldwide pandemic of H1N1 influenza, salmonellosis, West Nile virus, plague, and interestingly, emerging coronaviruses, SARS, MERS, but not SARS-CoV-2 were on that list. Rabies, brucellosis, both on the Alaska list, and finally Lyme disease, but that's not present here in Alaska. So our next steps were to develop action plans for each of these conditions or for the overall list. And we spent some time on this in the workshop on day 2, and we broke it into 4 areas: laboratory and surveillance activities, workforce development and outreach, outbreak response and preparedness activities, and One Health coordination. And we included some of the work done on workshop 2 in a report that was approved by the participants and will be made available on the CDC One Health website soon. So in conclusion, we found that the zoonotic disease prioritization workshop was a good learning and resource relationship building experience. We were able to get beyond the individual and organizational priorities and really get into some meaty issues about these zoonoses and come up with a list I think that was quite workable. And we were successful. We produced a priority list in 2 days of a lot of work, but came up with something that I think will help set a standard for the time to come. I think for these workshops, success is dependent upon really good preparation, having good discussion leaders, and we were fortunate to have several folks from the CDC who were able to help us through this process who've done these workshops before. Bringing committed stakeholders is really important, and we had a great group with this Alaska group, really engaged, involved, wanted to hear from each other and share ideas. And then I think another important part of success is planning for the follow-up. If I could do this again, I would get additional funding to bring more stakeholders, especially from rural Alaska. We held our meeting in Fairbanks just prior to a One Health meeting, thinking that might attract people who could travel. But this is often difficult for folks. More funding could have brought more stakeholders and could have broadened the discussion. I also would have gotten additional funding to support follow-up meetings or events to develop the action plans. I think this would be really important to carry through the activities and make sure that the list of zoonoses could be put into play. I'd also consider organizing circumpolar zoonosis prioritization workshops, perhaps 2 of them for the circumpolar north, one involving North America and Eastern Russia. So this might involve groups from Russia, Canada, and the US. And another that would involve Finno-Scandinavian participants and those from Western Russia, just because of the regional differences in zoonoses. And I think both of those would be really interesting conversations and could help international collaboration. CDC has shown interest in supporting workshops like this for the Arctic. They've done this in other international settings, and it may be something for Arctic participants to consider. The other thing I would do is I would wait until after the zoonotic disease pandemic has ended. Our work following this workshop was severely disrupted by COVID, as I'm sure everybody else's was. So I want to thank the workshop organizers and the people who helped put together the zoonotic disease workshop from the UAF One Health program, the state of Alaska, and the US Centers for Disease Control and Prevention. Here's my contact information and contact information for 2 folks at the CDC who could help organize subsequent workshops. Thanks for your attention. I really appreciate it, and I'm happy to take any questions.